Welcome to the Sand Science Experience, your source for the most current and impactful research and preprints in science. Hey, if you enjoyed this deep dive, please do hit that like button and subscribe. It genuinely helps support us and lets us keep bringing you these explorations. So today we're diving into something really fascinating. It's this connection, this surprising link between how our brains age and, well, the health of our entire body. We're looking at a new discovery in the fruit fly, Drosophila, and how just one tiny protein in its brain might actually hold a key to slowing down aging systemically across the whole body. Our mission here is to unpack exactly how that brain-body link could work.
It's pretty remarkable stuff, really, because it suggests that change is happening just, inside the brain can ripple outwards and affect aging everywhere else. It kind of challenges how we think about targeting aging.
Okay, let's get into it. I mean, we all know aging affects the brain. It's complex. Things like learning, memory, attention, making decisions, even just coordination. They can all get impaired. And of course, age is like the biggest risk factor for neurodegenerative diseases, Alzheimer's, Parkinson's. It's a huge health challenge globally.
Absolutely. And when scientists look closer at the cellular level of brain aging, one thing that keeps popping up is mitochondrial dysfunction. You probably remember mitochondria as the powerhouses of the cell. They generate most of the energy.
Right, the energy factories.
Exactly. And neurons, brain cells, they are really vulnerable when these powerhouses start to fail. See, unlike lots of other cells, neurons don't really divide or replace themselves easily. Damage just builds up. Plus, they have incredibly high energy needs. They rely almost totally on this one energy system, oxidative phosphorylation, just to function.
So when these power plants get old or damaged, they just sit there, clogging things up, like broken machines.
That's a great way to put it. And just like you'd want to recycle old machines, our cells actually have this built-in recycling system. It's called autophagy.
Autophagy, self-eating, right?
Precisely. It breaks down and clears out junk. And there's a special kind, mitophagy, which specifically targets those damaged mitochondria for removal. But studies show that as we age, this crucial mitophagy process, well, it tends to slow down. It becomes less efficient.
Okay, so the cleanup crew gets sluggish, old mitochondria pile up, and the brain's energy supply dwindles. Got it.
Yeah.
But this new research, it brings in this protein, BCL2 interacting protein 3, BNIP3.
BNIP3, yeah.
What's the deal with it? I heard its role was a bit murky.
That's right. BNIP3 sits on the outer membrane of mitochondria, and yeah, its function seemed a bit contradictory in the past, sometimes linked to cell death, sometimes survival. This Drosophila study really helps clarify things in the context of aging. So first, the researchers saw this really obvious buildup of dysfunctional mitochondria in the brains of just normal aged flies. Then came the key experiment. They specifically boosted BNIP3 levels, but only in the adult nervous system of the flies.
Ah, okay, so they targeted the brain cells in these older flies. What happened then when they flipped that BNIP3 switch just in the neurons?
Well, the effects were pretty dramatic. Boosting BNIP3 just in the neurons basically kickstarted mitophagy again. It cleared out those dysfunctional mitochondria.
Wow. And
it wasn't just cleaning up, it actually improved the brain's energy situation. They measured better mitochondrial membrane potential, and it reduced those damaging reactive oxygen species, ROS. It even seemed to be neuroprotective. It helps counteract the age-related loss of neurons and reduce the number of cells undergoing programmed death, apoptosis in the brain.
So it really reinforces BNIP3's job as a mitophagy receptor, flagging the bad mitochondria for removal.
Exactly. It directly links BNIP3 to that cleanup process.
Okay, brain benefits make sense.
Mm-hmm.
But here's where it gets, like you said, really interesting, the systemic aging part. Mm-hmm. How on earth does fiddling with brain cells affect the whole fly's aging? This cell non-autonomous effect thing.
Right. That's the truly mind-bending part. What they found was that boosting BNIP3 just in the neurons had these profound effects way beyond the brain. Crucially, it significantly increased the fly's overall longevity. They lived longer.
Longer lives.
But not just longer, healthier lives. Their health span improved. They saw less decline in their movement, better climbing ability, more spontaneous activity during the day. Basically, they stayed active longer.
So a healthier brain meant a longer and more vibrant life overall.
Exactly. And what's really promising is that they saw these benefits even when they started the BNIP3 boost midlife. That suggests potential for interventions later on.
That is promising. You mentioned effects in other organs, too.
Yes, quite clearly. In the muscles, the flight muscles, they saw smaller, healthier mitochondria and fewer of those age-related protein clumps. And in the gut, the intestine, neuronal BNIP3, reduced problems like stem cell overgrowth and mitochondrial buildup. It even delayed the breakdown of the intestinal barrier.
The Smurf assay thing, where they turn blue if the gut leaks.
That's the one. Preventing that blue leakage is a really good sign of healthy gut lining. So yeah, effects on muscle, gut, all from the brain.
It sounds almost like the brain is acting as a master controller for aging throughout the body. But, science is rarely that simple. Was it always beneficial? Or were there catches?
Yes, critical point. Two big things. First, all these benefits in the brain, lifespan, healthspan, muscle, gut, they were totally dependent on that autophagy pathway working. If they blocked autophagy in the neurons using a gene called ATG1, then boosting BNIP3 did nothing. The benefits vanished.
Okay, so autophagy is essential for BNIP3 to work its magic.
Absolutely. And the second point about precision, it turns out BNIP3 was only beneficial when boosted specifically in neurons.
Only in neurons?
Yeah, when they tried boosting it everywhere in the body, or just in muscle, or just in the intestine, it actually shortened the fly's lifespans.
Whoa, okay. So more isn't better, and location is everything.
Exactly. It really highlights that targeting is crucial. It's like needing a very specific key for a very specific lock, not just trying to force it open. So summing up, this research really points to neuronal BNIP3 as a potentially powerful target. Upregulating it just in neurons fights mitochondrial issues in the brain, and through this fascinating inter-organ communication, extends healthy lifespan systemically in the fly. And it underscores just how vital mitophagy is in this whole process.
It really makes you think, doesn't it? If tweaking something so specific in the brain cells of a tiny fly has such a massive impact on its whole body's aging, what could that mean for us, for human aging? Could focusing on brain health, maybe with really targeted interventions, be a way to achieve not just longer lives, but, healthier, more active ones too? Something to ponder. Well, that was a truly fascinating journey into the brain's unexpected role in how the whole body ages. If this deep dive got you thinking, please show your support. Hit that like button and subscribe. It really does help the Science Experience keep bringing you these kinds of discoveries. Thanks for joining us.